So the next topic is peptide bonds, and peptide bond is a bond using which the amino acids are linked together. So how it is formed is this is one amino acid and this is the other amino acid. They combine one carboxyl group of one amino acid combines with the amino group of the other amino acid, and one water molecule is released, forming a peptide bond, which is C double bond O NH. So molecules are referred to as polypeptides. They have a molecular weight of below 10,000 Daltons, while proteins have a molecular weight which is higher than 10,000 Daltons. And we also know that average molecular weight of an amino acid is 110 Daltons. Why? Because one H2O is always removed upon the formation of a peptide bond. So we subtract the molecular weight of water from it. So average molecular weight of amino acid is usually taken as 110. And the application of this particular thing is in finding out the number of amino acid residues that are present in a protein. You calculate the number of amino acid residues present in a protein using the formula wherein you divide the molecular weight of proteins by 110 Dalton, basically dividing the molecular weight of a given protein by the molecular weight of one amino acid. So for example, if we have a protein whose molecular weight is 50 kilodaltons, so to find out the number of amino acid residues in it, we divide 50 kilodaltons by 110, and we obviously convert kilodaltons into daltons. 1 kilodalton contains 10 to the power 3 daltons. So we get the final approximate number of amino acid residues in that particular protein. So also, peptide bond is a rigid and planar structure. It contains a partial double bond structure at this CONH region, and the carbon bond gives the value of psi, and carbon and nitrogen bond gives the value of phi. So the angle between carbon is psi, and between the carbon and this thing N is phi. These are called as dihedral angles, and on the basis of these dihedral angles we produce the Ramachandran plot. What exactly is a Ramachandran plot? Basically, Ramachandran plot is a plot of allowed and non-allowed structures of amino acids, and they are created by the values of the dihedral angle, that is phi and psi. So on the y-axis there are values of psi, while on the x-axis phi values are there along with the angles. So if we divide it in 4 quadrants, you can see both positive values and the values of those particular amino acids. So dividing the graph into 4 quadrants, we get the values or the allowed regions of particular type of amino acid structure. Basically, in the first, if we place, this is the first quadrant, this is the second quadrant, this is the third quadrant, and this is the fourth quadrant, we get to know that in the first quadrant only left-handed alpha helical structures are present. In the second quadrant, we have parallel beta sheets, anti-parallel beta sheets, right twisted beta sheets, and uh, collagen triple helix. So these are the allowed region in the second quadrant. In the third quadrant, we have all right-handed alpha helices, and if any of the amino acid fall in the fourth quadrant, it will be the disallowed region. So it won't be allowed, the structure won't be formed. So glycine, among the amino acid, glycine is the amino acid that shows the maximum combinations of the angles, the dihedral angle, that is phi and psi, and glycine acquires the maximum area in the Ramachandran plot, whereas proline, it shows the least amount of combination of dihedral angles phi and psi, and it acquires the minimum area. Moving on to the structure of proteins, there are different structures of proteins, starting from a primary structure, which is basically just the sequence of amino acids. The structure of amino acids or structure of proteins are determined using different methods, using different kinds of techniques. So the reagents, one is the Sanger Sanger method or Sanger method. Other one is the Edmond degradation method. So in the Sanger method, Sanger reagent is used, which is the fluorodinitrobenzene, and in Edmond degradation, phenyl isothiocyanate, which is also called PITC, this is used in the sequencing and finding the sequence of amino acid for the primary structure of the protein. In the secondary structure, the polypeptide chain is folded into alpha helix or beta sheet or beta turn in any of the three. The tertiary structure of a protein, in which the secondary structure is further folded, and it leads to a formation of tertiary structure. These are the tertiary structure is the one which is important for the biological activity of the protein, and the tertiary structure are stabilized by the disulfide bond as well as the hydrophobic interaction. The hydrophobic interaction in the tertiary structure, they play a major role in stabilizing the tertiary structure. The quaternary structure of protein consists of subunits, multiple subunits. So, for example, hemoglobin has a quaternary structure in which there are two alpha subunits and two beta subunits. And to each of the subunit, so basically to one hemoglobin molecule, four oxygen molecules are attached. Basically, one oxygen molecule attaches to each of the subunit. And normal hemoglobin levels is 12 to 16 grams per deciliter. This is the oxygen dissociation curve, which shows the Bohr's effect. In oxygen dissociation curve, the percentage saturation and the partial pressure of oxygen, it is plotted against each other. Bohr effect is basically when the curve of the oxygen dissociation shifts to right under the conditions when the partial pressure of carbon dioxide increases, partial pressure of oxygen decreases, there's an increase in the proton content, or the pH decreases and the temperature increases, or there is an increase in 2,3 bisphosphoglycerate. Going on to the details of secondary structure, first one we have is the alpha helix. So alpha helix is basically when the polypeptide backbone is tightly wound around the imaginary axis, and that axis is drawn longitudinally. Basically, this is the imaginary axis around which the polypeptide chain is rotated or wound helically, and the R groups, they protrude towards the outside of the helical backbone. Every turn in this helix has 3.6 amino acid residue. There is intramolecular hydrogen bonding, and intramolecular hydrogen bonding, it is different for different kinds of helices, but here in this case there is intramolecular edge bonding between the I and I plus 4, basically carboxyl group of one residue with the amino group of every fourth residue present in that helix. There are 36 amino acids per 10 turns, and 5.4 angstrom is the pitch. Helical twist of the alpha helix is always right-handed in all proteins. Basically, in alpha keratin, the alpha helix is the predominant structure, and the alpha helix, the maximum and optimal use of hydrogen bond internal, basically intramolecular hydrogen bond occurs. The constraint on formation of alpha helix is the presence of proline or glycine residues. Basically, if proline or glycine residues are present, alpha helix formation is difficult. Second structure is the beta sheets. They form intermolecular edge bonding. So there are two polypeptide chains, and the two polypeptide chains, within the two polypeptide chains, there will be intermolecular hydrogen bonding between the carboxyl as well as the amino group. As you can see in the diagram, there are two types of beta sheets. One is the parallel beta sheet and the other one is the anti-parallel beta sheet. Basically, in parallel beta sheets, the orientation of both the polypeptide chain is in the same direction, basically either from N terminal to C terminal. Both of the chains will have the N terminal on the top and the C terminal at the bottom. So this will be the parallel beta sheet. But in case the opposite terminals are present, like N terminal is at the top and C terminal is at the bottom, and in the second chain, C terminal is at the top and the N terminal is at the bottom, that will be the anti-parallel beta sheets. So between two amino acids, the distance is 6.5 angstrom in parallel beta sheets and 7 angstroms in the anti-parallel beta sheets. So in beta conformation, the polypeptide chain are extended in a zigzag structure as opposed to the helical structure, and they resemble a series of plate. The zigzag polypeptide chains, when they are arranged side to side, a plate-like structure is formed. That's why the arrangement of uh, this beta sheets, that's why it's called beta sheets, because they form like a plate, and the hydrogen bonding is between the adjacent segments of the polypeptide chain. When two or more beta sheets are layered close together within a protein, the R group of the amino acid residues on the touching surfaces must be relatively small. So basically, when they are close, the side chains should be small so that proper structure is formed. The smallest amino acids which are there are the glycine and alanine. Beta carotene, such as the silk fibrion or the fibrions of spider web, they have high content of glycine and alanine. The two smallest amino acids, jinka side chain is the smallest. So in silk fibrion, the glycine and alanine alternate over a large part of sequence. So they are the major structure in the beta carotene. Next structure is the beta turn. Beta turn is just the connecting element that link different structures, different secondary structure of proteins, such as alpha helix or beta conformation. The beta turn is a 180-degree turn, and it involves four amino acid residues. The two amino acid residues in this beta turn, they do not participate in any kind of inter residue hydrogen bonding, and mostly in beta turns, glycine and proline are present. So glycine makes the turn flexible while proline makes it tight, so it keeps the things on place. Then there's a gamma turn, and in gamma turn there is hydrogen bonding between the first and third residue, and the gamma turn is very rarely found. So moving on to the proteins, the different kinds of proteins. There are two major types of proteins. First one is the fibrous protein, and the second one is the globular protein. In fibrous protein, they have polypeptide chains which are arranged in long strands or sheets, and they usually contain a single type of secondary structure. The examples of fibrous proteins are alpha keratin, collagen, silk fibrion. Whereas if we talk about globular proteins, they have polypeptide chains and they are folded into spherical or globular shape, and they might have different types of secondary structure, or else like subunits, they have subunits. For example, the examples of globular protein are immunoglobulins, transport proteins, membrane proteins, regulatory proteins, or certain enzymes. Talking about examples of fibrous proteins, we'll be studying about alpha keratin, collagen, and silk fibrion. The fibrous proteins, all the fibrous proteins are insoluble in water. And why are they insoluble in water? Because there are high concentration of hydrophobic amino acids both in the interior surface of the fibrous protein as well as on the surface. The first one which we talk about, the first fibrous protein which we talk about is the alpha keratin. And alpha keratin basically is a protein that has evolved for strength, and it's a part of a family called intermediate filament proteins, and alpha keratin is a right-handed alpha helix. If two strands of alpha keratin, they are oriented in parallel, wrapped around each other, they form a super twisted coiled coil, and its helical part is self sorry, its helical path is left-handed. Basically, two alpha keratin strands oriented in parallel, wrapped around each other. They form a super twisted coiled coil, and which is basically helically left-handed. The alpha keratin is rich in the amino in all the hydrophobic amino acid, which is basically alanine, valine, leucine, isoleucine, phenylalanine, and methionine. And alpha keratin structure is stabilized using the cross links of disulfide bonds. Next fibrous protein is basically collagen, which is the most abundant protein in animal world. It's obviously again for strength. It is found in connective tissue in the body such as tendons, cartilage, organic matrix of bone, and cornea of the eye. Basically, one fun fact: cornea of the eye is the part which of the eye which is donated during eye donation, since there is no blood supply in cornea. Coming back to collagen, it is a left-handed helix. Three amino acid residues are present per turn, and there are three separate polypeptides in collagen. They are the alpha chains, which are super twisted about each other, and the alpha chain, when they're super twisted, it's right-handed. The collagen contains 35% glycine, 11% alanine, 21% proline, and a 4-hydroxyproline. So these are the four amino acids in the ratios that are given in present in collagen. Hydroxylation of proline occurs in the presence of vitamin C, or which is also known as ascorbic acid. A tripeptide unit is repeated multiple times in collagen. The tripeptide unit is glycine, proline, and 4-hydroxyproline. Glycine, proline, and 4-hydroxyproline is the tripeptide unit, which is mostly repeated multiple times in collagen. There's a syndrome called Ehlers-Danlos syndrome, wherein the collagen is not formed properly and it is characterized by loose joints. Moving on to the next kind of fibrous protein, we have the silk fibrion, which is basically anti-parallel beta sheets, and they are produced. They are part of a protein of silk produced by insects and spiders. They are predominantly in beta configuration or conformation. They are rich in alanine and glycine, due to which a close packaging of beta sheet occurs, and there is an interlock arrangement of the side chains. The silk doesn't stretch because the beta conformation is already highly extended. However, the structure is flexible because the sheets are held together by numerous weak interactions rather than by the covalent bonds such as disulfide bonds in alpha keratin. So basically in silk there are numerous weak interactions due to which the structure is flexible, but the sheets are also held together. Structure there is called motif, which is also called a super secondary structure or a super secondary fold. It's basically a recognizable folding pattern which involves two or more elements of the secondary structure between them. For example, the examples of motif are beta, alpha, beta. Three kinds of secondary structure are connected in this: beta barrel, and then there's zinc finger motif, which is involved in a nuclear localization signal, and this zinc finger motif is rich in cysteine and histidine. Coming on to domain, it's a part of polypeptide chain that's independently stable or could move as a single entity with respect to the entire protein. There's a technique which is used to find the secondary structure of proteins. So circular dichroism spectroscopy is the technique using which we find the secondary structure of proteins. Circular dichroism spectroscopy is used to find the secondary structure of protein. How it's done is when the chromophores of amides of polypeptide backbone of proteins are aligned in arrays, their optical transitions are shifted or split into multiple transitions due to excitation. And CD spectra, the circular dichroism spectra, is created. So this is what a spectra of proteins secondary structure looks like. Alpha helix is this curve. This curve shows the CD spectra of alpha helix. This is anti-parallel beta sheets, and the random coil is this one from, sorry, yellow. Okay, so how you do it is basically the alpha helical structure. They have negative bands at 222 nanometer and 208 nanometer. They have positive bands at 193 nanometer. In case of anti-parallel beta sheets, negative band is shown produced at 218 nanometer, around 218 nanometer, while the positive bands at 195 nanometer. And in case of disordered proteins, whose secondary structure is not as ordered as in case of alpha helical or anti-parallel beta sheet, there is a very low ellipticity above 210 nanometer, and they show negative bands near 195 nanometers. So these are the wavelengths at which different secondary structure of protein produce the CD spectrum. And this is the spectra that is produced for different secondary structures. So in case of CD spectra, there's an unequal absorption of left-handed and right-handed polarized lights. What are the polarized lights? Polarized lights are basically light waves in which the vibrations occur only in a single plane. So when asymmetric molecule interacts with light, they may absorb light in the left-handed or the right-handed, and they circularly polarize the light to different extents, either in the left-handed direction or the right-handed direction. And they have different indices of refraction for those two waves, left and right. So we calculate the resultant traces of the ellipse, the resultant traces, which is the basically the sum of vectors of ER and EL. So in the right direction and in the left direction. Index of refraction is basically a measure of the bending of ray of light when passing from one medium to another. Okay. Next part is that of hydrolytic enzymes. Hydrolytic enzymes are the enzymes which break the polypeptide chain from different locations, or they they cleave the polypeptide chains from certain points in the chain from certain amino acids. The hydrolytic enzymes include trypsin, chymotrypsin, carboxypeptidase B, cyanogen bromide. These are the cyanogen bromide is the chemical. So this is an example of a polypeptide chain which shows the different cleavage sites for different kinds of hydrolytic enzymes. Talking about trypsin, which is represented by the letter T, the alphabet T, it cuts after the amino acid lysine and arginine. However, the conditions required for its cleavage is that proline shouldn't be present right after lysine or arginine. So if proline is present right after lysine and arginine, trypsin won't cleave the polypeptide chain. So it cuts after lysine and arginine provided proline is not present, and trypsin does not act at the terminuses. Basically, neither at the carboxyl terminus nor at the amino terminus. The next hydrolytic enzyme is the chymotrypsin. The chymotrypsin cuts after the aromatic amino acids. The aromatic amino acids are phenylalanine, tryptophan, and tyrosine. It also does not act at the terminus. Again, the condition is that proline should not be present right after them. Carboxypeptidase B is another hydrolytic enzyme which cuts before lysine and arginine, and it acts only at the terminus. Then there is cyanogen bromide. It cuts after methionine. So this is it for the hydrolytic enzymes. Now the next topic in this is the precipitation of proteins. First one is the ammonium sulfate precipitation, in which ammonium sulfate is used to precipitate the proteins. How does it precipitate the proteins? Basically, it exposes the hydrophobic amino acids of the protein, which leads to the hydrophobic interaction, and a missense out of a structure is formed, and it leads to the precipitation of proteins. So higher the number of hydrophobic residues, faster is the precipitation of proteins. As you can see in the diagram, there's this structure, and the hydrophobic part is formed in the interior of this micellar structure, and whereas the hydrophilic parts are at the outer side. Such structures are formed which precipitate after the action of ammonium sulfate. Molten globule is basically a compact, partially folded conformation of proteins which have near native secondary structure, and they have a little detectable tertiary structure. They have increased solvent exposed hydrophobic surface area relative to the native structure. The molten globule contains native-like secondary structure content. However, they do not have tightly packed protein interior. Under the conditions are low pH and high salt concentration. And the molten globule also includes various types of partially folded proteins, uh, protein ka states, which are found under the mildly denaturing condition, and the mildly denaturing conditions are that of low pH, generally 2, or some mild denaturant might be present, or a high temperature. So the precursors for different amino acids are written here. Precursor for arginine, glutamine, glutamate, proline is alpha ketoglutarate. So basically, glutamate is formed from alpha ketoglutarate, and from glutamate, either glutamine, proline, and arginine are formed. From 3-phosphoglycerate, serine is formed, from which glycine and cysteine are formed. Aspartate, aspargine, methionine, threonine, lysine, isoleucine, they are derived from oxaloacetic acetate. So this is how it's formed. Oxaloacetate forms aspartate, which forms asparagine, methionine, threonine, and lysine. From threonine, you get isoleucine. Alanine, valine, and leucine and isoleucine are derived from pyruvate. So this is it for now. Hope it helps. Thank you so much.